Thank you all for giving George and I the chance to reinforce what we're doing with the How Often effort. This is something that George and I presented a couple months back, and we wanted to encourage the community to participate. We're going to use this session as an opportunity to detail exactly how you all can make immediate contributions. And then hopefully, as we go through this, I'm going to present for a little bit, and Gowtham's going to remind you all about how to use the phenotype library. And then we're hoping to actually use the last half of the session to do breakout brainstorms about how often is going to mean to you. So just a quick reminder, back on June 20th, George presented about the genesis of the How Often project. This is a large-scale incidence characterization initiative whereby many years back, we had this idea that it would be nice if we could compute the incidence of all things against all things and make that publicly available. And we talked about the learnings of our community about how to compute incidence rates in a reliable way. And we also talked about the progress that we've made as a community in developing open source solutions and actually having very meaningful clinical applications for specific questions. But to build off of that, we see a really big opportunity, and one of the strategic priorities that George and I have laid out over the last several months is the opportunity for us to come together as a community and as a data network to execute a large-scale analysis that will produce incidence of a large set of target exposures or target cohorts against a large set of outcomes. And so at that presentation back a couple months ago, we motivated this by what Craig just showed, which is at our global symposium, we are going to be taking a full day as a full community to focus on how often. And this is something that George and I feel very strongly about, that we want to see more community-wide efforts to try to advance our shared cause of evidence generation. And so we thought, what better way to do that than to bring everybody together to focus together as one large community on one large effort that we know we can make a meaningful difference on. But of course, we're not going to solve a major healthcare problem in only 6 hours together. We've got to do some homework ahead of time, and we're going to certainly have follow-up afterwards. But what I highlighted a couple months ago was exactly what this How Often community effort was going to be. And effectively, the process that we are following is, in order for us to compute the incidence of a large collection of target cohorts against a large collection of outcome cohorts, what we need to be able to do is first generate phenotypes that will represent those target cohorts and those outcome cohorts. From there, we will be able to design a protocol and implement an analysis specification whereby we detail each of those cohorts that we are going to use, detail the way in which we will conduct a statistical characterization analysis to generate an incidence calculation, in detail how we are going to disseminate that information. The reason why we want to have that protocol and analysis specification done is so that we can do two things in parallel. The first is that we can implement not only the package that will generate the results, but by knowing what those results will be, we can work together as a community to develop an interface that will allow for exploration of those large-scale results. And also, for those of you who have access to data that may need to go through some sort of governance process to have permission to execute an analysis, we want to give you sufficient lead time to be able to get those permissions and come to the symposium and be able to execute this study and share back the results. Ultimately, when we conduct this analysis, we will be generating a very large collection, the world's largest collection of incidence rates for a large set of target and outcome cohorts. And there's going to be a lot of opportunity for us to collaborate as a community to interpret those results. And from there, we can both publish methodologic findings about what was it like to compute incidence across a distributed data network with a wide array of different sources and geographies. And we can also publish some of the clinical implications of what we learn from some of the questions that may be raised. Now, I've mentioned, and as I mentioned a couple months ago, as we detailed this, when we talk about target cohorts, we can think about that as the denominator population at risk. If we're going to compute an incidence of something, we're usually talking about some population that's at risk, and amongst those people, some events that take place amongst them. And there's a wide array of different target cohorts that we could consider studying. What I highlighted a couple months ago is we could think about just a general population, like everybody who's in a database on a given date. We can think about specific indications, like a disease cohort, people with diabetes. We could also think about people who are exposed to a particular medical intervention. So, people with a drug record of a GLP-1, for example. Really, any target cohort just represents a population that's well-defined for some duration of time, for which we know when they enter a cohort and then we know when they leave, and we can use that cohort to represent a population at risk. Now, the outcome cohorts represent the events that we would want to look at. So given that there's some group of people in a target cohort, we'd like to know which amongst those individuals go on to experience some sort of event. And these could be things like the adverse events of special interest, of which our community has done a lot of good work in phenotyping to support COVID vaccine safety surveillance efforts. It could be the broader set of designated medical events that the European Medicine Agency actively monitors for medical products. It could be outcomes that are, particular relevance for specific indications. It could be side effects of drugs. It really could be any type of observed event in a given data set. And given that we have a defined list of target cohorts and a defined list of outcome cohorts, our goal with our HowOften effort is to make it possible for us to computationally efficiently compute every target versus every outcome to compute the incidence, and further stratify that by data source, by age, by sex, and by calendar year. Now, where you all come in is that for this to be a community effort, there's a wide array of things that you can be doing to participate in this effort. While George and I are particularly motivated by seeing this get done, we recognize that we absolutely cannot do this by ourselves. Huge area of need, and where we're going to actually focus a lot of today's conversation on, is phenotypers. For us to do this, it's all predicated on the fact that we have target cohorts and outcome cohorts ready and available. We also need folks who are interested in developing the analysis package. We have existing standardized tools within the OHDSI toolkit that we will be making use of, but we of course have to wrap that into a network study that we can run across our community. If you're out there and you like data visualization and thinking about how to make interactive exploration of data, we certainly can use your help as we think through that work. We're certainly looking for data partners all around the world to be able to participate and contribute by executing the analysis and sharing back results. And we're looking for methods researchers who are interested in thinking about this problem of how to produce a reliable incidence estimate across the data network. And we're also interested in clinical researchers who are interested in, given a set of incidents, what have we learned that maybe fills the evidence gaps that are in the literature. So this is what I showcased a couple months ago, and what today we're going to do is try to drill down a little bit further into why we need your help with identifying targets for the phenotypes, and having your help for the next month or so in building those phenotypes and contributing them to our phenotype library. So I guess a couple months ago we talked about that there's some pre-symposium work that needs to be done. George and the team are focused on trying to draft a protocol that will allow for approval from data partners, and that's something that we hope to be able to make available in the near future. Part of that protocol, though, needs to enumerate the list of phenotypes that are going to be used, and that's where we really need all of your support to make sure that we have that full set of phenotypes available. And we will have an analysis package that implements that protocol with all the phenotypes available prior to the symposium. What's going to happen at the symposium is that we are going to work together as a community to run this analysis package across the OHDSI network. So for those of you who are data partners who may need some help with getting something installed and configured and run in your environment, we'll certainly be there to help. But we'll also work together with the results that already exist to be able to explore those results, continue to work on the development of the interface, consider the methodologic questions about how do we trust this evidence and prove that the evidence is trustworthy, as well as focus on the clinical questions about what we've learned and how that contributes to the current evidence that's out there. Now, I would just want to highlight that when we talk about incidence, we're really just talking about one specific narrow use case in our OHDSI set of analytic cases. We talk about characterization, estimation, and prediction, and really characterizing incidence is just one facet of this. So our emphasis on this isn't to say that this is all we should be doing, but rather to say all of the types of analysis use cases that we've been talking about as community are valid and relevant. However, we think we can make really good progress if we were to just focus on one, here incidence, because there's a standardized way that we can represent what an incidence is supposed to be, and we can standardize our analytics and then standardize our way to disseminate that evidence. And that's really where we're going to focus on as a community over the next couple months. So to highlight, I guess, three very narrow efforts that have been done in the community where incidents proved to be important. The work that we had done to support our colleagues at the FDA and the EMA around the incidence of adverse events of special interest for COVID vaccines is a notable example where within our community, we've now published multiple papers highlighting the idea that across a large international data network, we could compute incidence rates for a collection of 20 outcomes that were the object of potential concern associated with the COVID vaccines, and that we could contextualize those background rates so that they could be used when regulators and vaccine manufacturers were monitoring the safety of those vaccines, comparing that data to spontaneous data, as well as to consider what methodologic issues were raised when thinking about how to appropriately use those incidents. And I'm really proud of the work that our community has done in contributing this evidence out there to support regulatory decisions, as well as to kind of advance the science of incidence estimation across the data network, as well as to demonstrate how we as a community could do a large work. Erica's paper that's been highlighted multiple times, illustrating how 26 data partners were able to come together to generate evidence, is a nice demonstration of how as a community we can work together. A more recent motivation for this is the work that Ben Hamlin presented on August 1st on our community call, where we talked about quality measure development. And one of the things that Ben highlighted is that really, at the end of the day, these digital quality measures that are used actively as a primary mechanism of monitoring healthcare performance in the United States really are just nothing more than an incidence proportion. They are basically a denominator cohort of patients at risk and a numerator cohort of people who experience some desired rule. And so if you are interested by the quality improvement area, thinking about that in the context of how often should be right up your alley. And there's, of course, a defined set of quality metrics that exist that we could implement, as Ben highlighted as one of the efforts of the HL7 September Connect-a-thon. The other one I'd like to highlight is the work that we've done as a community with the Sisyphus Challenge, where we've identified multiple questions of interest. And I'll just highlight to reinforce that if we took a look back at Cindy's study, looking at the causal effects of anti-VEGF exposure on the risk of end-stage renal disease, what we actually see is that we actually computed incidence and characterized the incidence, not just of the exposed population, but also of the underlying indication. And so what I've shown here is a screenshot of our standardized application that is using our strategist platform. And here one could just select the indication, here people with blinding diseases, select the outcome, end-stage renal disease, and they could actually see the incidence for a given database presented directly through our interface. And in fact, this incidence information is something that I expect Cindy and the team is going to use in their anti-VEGF paper. I know that Jack and Nicole and Chan and Jung Ho and the team that focused on fluoroquinolones and aortic aneurysms is additionally focused on this. And then Thamir's other study is specifically focused on the incidence of PML in biologics actually being the targeted outcome of the study itself. And so these are cases where somebody had an idea of a particular target cohort, a particular outcome, and wanted to have an incidence for a motivating reason that really has some direct merit. Now, what I want to just augment to that conversation is why George and I are really focused on this idea that we need to do large-scale incidence characterization, to go beyond answering questions one at a time and to think about the problem at large. And to motivate that, I'm just going to try to illustrate two ideas. First, I'm going to show this matrix that I produced many years back, but I think it's still salient to this point. Imagine we had a matrix that represented all drugs on the y-axis and all outcomes on the x-axis. Now, if I take a more recent look through the data, if I took one of my large U.S. claims databases that I have access to, I can identify that there are over 2,000 active ingredients that are FDA-approved drugs that are prescribed to at least one patient in a database. So think of that as the dimensionality of this y-axis is 2,000 long. And if I were to try to define the dimension of all outcomes, a very crude and simple proxy could be, what if we just took each of the individual source codes that we had? And in a US claims database, there's over 50,000 distinct ICD-10-CM codes representing, according to the ICD vocabulary, distinct medical conditions that are observed for at least one patient in that same database. So what that actually means from a large-scale perspective is if there are 2,000 drugs and 50,000 outcomes, that means there's 100 million potential drug outcome relationships for which it would be very reasonable that some patient out there would want to understand, and I would assert deserves to know the answer to the question, how often does that outcome happen amongst patients who are exposed to that drug? And this basic idea that patients deserve to know how often something might happen if they're exposed to a drug was the original genesis of the original How Often project that we had kicked off many years ago. And in that version, our basic premise was to do code-based phenotypes. That is specifically to say, we'll just use all RX norm ingredients, we'll use all SNOMED concepts in our OMOP CDM, and we will just permute through that combination to produce an evidence base that potentially the community could explore. And I would assert that that's still a good opportunity for us to dust off that idea, implement it a little bit more efficiently and a little bit better, and implement. And George and his lab at Columbia are actively working to think about how to pursue, continue that idea and expand it even further. What we're talking about today, however, is something that I would say like even expands this universe even further. Because here I've defined the y-axis as drugs, and I've defined the outcomes as conditions. But in fact, the real opportunity in front of us is that we get to define whatever this matrix is, however we want. And so what I'm going to really primarily describe here is that I want us to think about, given that we can compute the incidence of any combination of any target versus any outcome, we get to define what this space is. So here I'm just kind of reimagining that matrix there to show that on the y-axis, any target cohort could be any denominator of a population at risk that you could come up with. And it doesn't need to be restricted to drugs. It could be drugs or conditions or procedures or measurements or the calendar years or some specific population that you're interested in. The main thing is simply that we just need to be able to define it as a cohort. And we can have as many of these cohorts as we potentially are interested in. Similarly, on the x-axis, we can talk about outcome cohorts, and these don't need to be code-based conditions. They don't have to just be a SNOMED term or an ICD code, but rather we can think about these as just the events that we want to observe as whether or not they occur within a population at risk. And so if we can define proper cohorts, those could be, the onset of a disease, but they could also be, the elevation of a laboratory value or the, initiation of a new treatment or people who go on to have a surgical procedure. Whatever is of salient interest to you as a researcher, to patients and clinicians who are looking for evidence, that's the target that we want to go. And the point here is that if we have, any number of N target cohorts and M outcome cohorts, the goal that we have is let's apply standardized analytics to consistently and reproducibly generate the incidence for every one of those N by M combinations, such that I can pick any target cohort that we've defined and any outcome cohort that I find, and I'm basically going to be answering the same fundamental clinical question, which is amongst patients who are in target cohort I, how many patients have experienced outcome J within some time horizon? So the idea of our how often effort is for us to create a standardized framework for which we can generate credible evidence answering this type of question across the full continuum of target and outcome cohorts that we may be interested in. Where we need your help is that we don't know what all those target cohorts and outcome cohorts are, and we want to be able to create those definitions so that we can make use of them. So how can you contribute now? Our primary focus right now, above all else, is that we need to create those target cohorts and those outcome cohorts. And what that tangibly means is to contribute them into the OHDSI phenotype library. And so I'm going to check to see if Gowtham, are you online?
Yeah, I'm on the call.
Great. So can I just turn it over to you to walk through how people can make a contribution to the phenotype library, and I'll drive your slides for you?
Oh, absolutely. Thank you, Patrick. So, thank you. As Patrick said, OHDSI Phenotype Library would be a conduit for the how often study. And most of you know that the OHDSI Phenotype Library has been around for some time. Currently, it is in version 3.1.6, 3.1.16.1. So this is the landing page of the OHDSI Phenotype Library. Can you go to the next slide, Patrick?
Yeah.
So let me first talk about what the OHDSI Phenotype Library is. So the OHDSI Phenotype Library is a catalog of computable phenotype algorithms. This is what we call as cohort definitions, and they are compatible with the OHDSI standard software. The unit of representation in the OHDSI Phenotype Library is a cohort definition. So this cohort definition could be a cohort JSON, which is a Cersei-defined phenotype model, or an SQL. And when you execute this using an OHDSI standard software on a person-level data set represented in OMA common data model, you get a cohort. The OHDSI Phenotype Library catalogs these cohort definitions. Now you can access this through the OHDSI GitHub repository using either a GitHub API or an R package that presents all the cohort definitions that are in the library. You can access it. There are some applications that are currently using the OHDSI Phenotype Library, like the data.ohdsi.org Phenotype Library, and also dash.ohdsi.org Phenotype Explorer. Can you go to the next slide? Now, the Phenotype Library follows certain principles. It is publicly accessible. It is version controlled. It is referenceable. So if you have a study and you would like to reference a cohort definition, you can do that. And importantly, it attempts to go towards the FAIR principle, which is findable, accessible, reproducible, and interoperable. For every cohort definition, which is the unit that is represented in the Phenotype Library, it catalogs the metadata. It attributes to the person that contributed the cohort definition. And it also promotes the discovery and reuse of these cohort definitions for scientific research. So these are the principles of the OHDSI Phenotype Library, available at github.com/ohdsi/phenotype library. Can you go to the next slide? Also, one important clarification. There is no gold standard. The OHDSI Phenotype Library allows for more than one cohort definition to be contributed for the same clinical idea. Now, so specifically, if you may see the Phenotype Library having a definition for small cell lung cancer, but you may think that your definition is different because it has some operating characteristics that you think is better than the previously submitted cohort definition. You can then submit that new cohort definition as an alternative for small cell lung cancer. The library by itself does not recommend a definition for, say, hypertension or small cell lung cancer. It catalogs the submission and the metadata. It allows for the user to make the decision based on the evaluation report that the submitter has submitted, and also the operating characteristics that may have been computed by running the cohort definition on person-level dataset. Now, there are very few exceptions, and if it's a logically identical cohort definition, for example, a cohort definition which produces an additional stat about gender, just because they want a gender-specific cohort definition. Minor variations are something that we try to avoid, but overall, as long as there is good enough, there is a difference in performance characteristics, we will accept the cohort definition to the OHDSI. Next slide. Now, the submission to the OHDSI Phenotype Library is straightforward. The first step is to create an OHDSI forum post. We have recommended a certain template. You follow with a title that says Phenotype Submission, and that's a prefix, followed by the name of your clinical idea, say acute hepatic failure. The reason for this is it makes it easy to search, and also you can then reference it and ask for another collaborator to review it for you. There is a more detailed guidance on how to submit it, but the submission process is through the Odyssey forum. Now, if at any time you find this cumbersome, you can always come to the Odyssey Phenotype Development Evaluation work group, and we can work with you on a custom way of submitting core definition to the Odyssey Phenotype library. Next slide. Now, once you start an Odyssey forum post, we ask you to submit the core definitions. This is usually a text file, so you can attach a text file into the Odyssey forum post. And if you have developed the core definition using your Atlas instance, you can copy the JSON, save it as a text file, attach the text file by uploading it to your forum post. You can submit more than one core definition, and as long as you give it a clear name, then it's easy to distinguish between them. Next slide. Now, if you submit core definition, we want to know what that core definition is so that it is findable. You can discover. So we want a name for it. We want a short name, maybe a long name that is more meaningful, so that others can discover your core definition. We want to provide attribution. So we would like to know who the contributor is. If there's one person or many person, a team of people that develop the core definition, we would like to know their name. The org IDs are better than full names because it's more computer friendly. So we would recommend using that. For every phenotype, we would like to know its clinical description. So if you are modeling a complex clinical idea of a person who developed hip pain after a surgery, we would like you to describe that clinical idea so that others know what is the target population that you have modeled in your core definition. Next slide. There is the idea of a logic description. So logic description is if you remember, if you know in a method section in a paper, we would describe in a less technical terms how we model this core definition. What the example could be: we require the persons to have an age of more than 18 years. We require them to have 2 doses of hydrochlorothiazide. That idea in non-technical terms is something that we call as logic description. And it helps us to understand and the rationale, the thinking behind your core definition. We also have an attribute which is called the recommended study applications. So if you think that this core definition is best suited for an outcome analysis, an outcome definition, or if you think it is something that could be used as a target, or if you have some idea that you would like to convey to a recipient of the core definition, we recommend you to post that in your forum post. And more importantly, we have something that we call as an assertion statement. Assertion statement is that you have developed this core definition and have executed that core definition on at least one person-level data source in an OMAP common data model format, and have had at least one person returned in that cohort. And that's important. And that's mainly to make sure that the core definition that has been built has been used in some form by the submitter. So these are the minimum requirements for core definition. And as long as you meet this minimum requirement, the core definition will be put into the Odyssey Phenotype library. Now, once it's in the Odyssey Phenotype library, you have the option of going through a peer review process. The peer review process is handled by the Odyssey Phenotype Development Evaluation Workgroup. And if you go through the peer review process, you get an additional level of flag of quality. There's other optional recommendations, and optional recommendations could be you could provide back to the community the output of your core diagnostics execution, the output of your fee evaluator execution, and those would also be something that will be captured by the Odyssey Phenotype library. And that's all I have about this.
Great, thank you, Gowtham. So so given that Gowtham has just nicely walked you through the phenotype library and the submission process, the assertion that we are going to make is for this HowOften Symposium activity, we are going to base our analysis exclusively on phenotypes that exist in the Odyssey Phenotype library. So if you go out to the Odyssey Phenotype library right now, you are going to see that there are many hundred phenotypes that have already been contributed. A relatively small proportion of those have actually gone through that peer review process that Gowtham detailed. And so not all of them have been fully evaluated and are necessarily ready for primetime use. But by having this standardized infrastructure in place with the phenotype library for us to be able to use as an input to our process, it will allow our HowOften analysis to use the phenotype library as its input and to be able to define the targets and outcomes that will be used in the analysis. So what that means is, very simplistically, if you want a cohort to be included in the HowOften experiment, you need to create a phenotype and it needs to get into the OHDSI phenotype library. While the symposium is not until October, of course, I mentioned we have a couple logical constraints here. One is that we need to develop the analysis package before the symposium. And second is that we need to finalize the protocol so those of you who are data partners who have governance restrictions can make sure that you get the appropriate approvals for participation. As such, we're asking that if you are interested in having a phenotype contributed to the HowOften effort, that you get your phenotype into the OHDSI phenotype library by September 15th. That's, I guess, more than a month from now, or a month from now, to get your phenotype developed and evaluated and contributed into the library so that we can consider pulling it into the HowOften effort. I'm using this purposely a little bit as a forcing function, in that we had a very productive phenotype February where Gowtham and Azza and many members of the community demonstrated how to develop phenotypes and how to evaluate them and how to work together as a community. But since then, we've actually seen that the number of contributions to the phenotype library has been relatively slow. And so we hope that this forcing function of the fact that you're not entering it just for the sake of entering it, but you're entering it because it will ultimately get directly input into a large-scale effort that you will then get to be a contributor and collaborator on. We hope that that will motivate more sharing from within our community. We're setting this deadline of a month, but recognize that the community is here to help you all. In addition, Gowtham and Azza host the Phenotype Development and Evaluation Workgroup meetings, which are on Fridays. And so you can join there if you've got questions, but also feel free to post on the forums, ask questions, and raise any concerns that you have about trying to get you to the finish line on September 15th. It'll be September 15th when George and I will sit down, look through the phenotype library, and identify those that folks are interested in using as targets and outcomes within the effort to be able to define that protocol. So if you're not in the phenotype library, you won't be considered for HowOften. If you are in the phenotype library, you will be considered. It's that simple. And so we hope that you will take this as an opportunity to use the next month to make contributions to be able to participate in this HowOften effort.